Dear biology aspirants, right now I have the electron transport system ETS. This subtopic will come under respiration in plants, and you will see here some functions of electron transport system, that is the ETS, also called electron transport system ETS, and oxidative phosphorylation is the key matter of for the discussion. The following steps in the respiratory process are to release and utilize the energy stored in NADH plus H and FADH2. This is accomplished when they are oxidized through the electron transport system, and the electrons are passed on to O2, resulting in the formation of H2O. The metabolic pathway through which the electron passes from one carrier to another is called the electron transport system. Figure 14.4 will provide you the information regarding what I have said, and it is present in the inner mitochondrial membrane. Electrons from NADH produced in the mitochondrial matrix during citric acid cycle and oxidized by an NADH dehydrogenase complex 1, and electrons are then transferred to uh, ubiquinone located within the inner membrane. Here you will see also receive reducing equivalents via FADH2 complex 2 that is generated during oxidation of succinate in the citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electrons to cytochrome C via cytochrome BC1 complex 3. Cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier to transfer of electrons between complex 3 and 4. Complex 4 refers to cytochrome C oxidase complex containing cytochromes A and A3 and 2 copper centers. When the electrons pass from one carrier to another via complex 1 to 5 in the electron transport chain, they are coupled to ATP synthase complex 5, and here you will see the number of ATP molecules. Synthesis depends on the nature of electron donor. The number of ATP molecules synthesis depends on the nature of electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while that of one molecule of FADH2 produces 2 molecules of ATP. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. Yet the presence of oxygen is vital, since it drives the whole process by removing hydrogen from the system. Oxygen acts as the final hydrogen acceptor, unlike photophosphorylation, where it is the light energy that is utilized for the production of proton gradient required for phosphorylation. In respiration, it is energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane linked ATP synthesis is explained by chemiosmotic hypothesis on in the earlier chapter. As mentioned earlier, the energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthesis complex 5. This complex consists of two major components of F1 and F0. Figure 14.5 will provide you the information what I'm saying. And F1 headpiece is a peripheral membrane protein complex and contains the site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein complex that forms the channel through which protons cross the inner membrane. The passage of protons through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, 2H plus passes through F0 from the intermembrane space to the matrix down. The electrochemical proton gradient what I have discussed is here. So next course of action, I will provide you the respiratory balance sheet, and here you will see the diagrammatic presentation of ATP synthesis in mitochondria. So students and aspirants, what I have said, what I saw it you do for your understanding. In next video, I will discuss lots more for the biological domain and biology subject, and I am going to give you the thanks for the watching this video. Thank you.